This is Laboratory 9 that deals with hemoglobin electrophoresis, and the objective is really to allow the students to become familiar with the technique of hemoglobin electrophoresis, which is a laboratory test that usually measures hemoglobin molecules and their variants in the blood, because there are several types of hemoglobin that is present normally in the adult, and there are variants to these, and we use electrophoresis to determine what are the variants present that may be causing disease, for example, like sickle cell anemia and so on. Okay. Hemoglobin consists of a protoporphyrin ring, as you remember, and it has 4 polypeptide chains. They're called globin chains, and these chains are made up of 2 beta chains and 2 alpha chains. Each normal hemoglobin molecule has several types of beta chains, or chains other than alpha chains. They all have alpha chains, but some have minor fractions normally of hemoglobin A2 and delta chains. So the hemoglobin A2 contains alpha chains and delta chains. So the major hemoglobin in the erythrocytes in the adult also contain a small amount of hemoglobin A2 and hemoglobin F, which is fetal hemoglobin. There are over 100 variants of hemoglobin now known, and in many cases they don't cause or exhibit any abnormality, and therefore they are completely silent. But there are some that can cause serious clinical effects, especially if the individual has the genes for the defect from both parents, that is a homozygous state. Hemoglobin disorders can be broadly classified into 2 general categories. You have those in which a quantitative defect in the production of the globin subunit. Quantitative defect may be the total absence of the subunit, the globin subunit, or marked reduction. And these are known as the thalassemia syndromes, which we discussed in lectures. And then there are those in which there is a structural defect in one of the globin subunits. And the principle of this electrophoresis is that hemoglobin is an alkaline pH and is negatively charged when it is subjected to electrophoresis, and therefore it will migrate from the cathode to the anode. And structural variants within the hemoglobin that you are measuring will have a change in charge on the surface of the hemoglobin molecule at alkaline pH, and therefore they will tend to separate on the acetate cellulose paper that we use. There are some limitations to this test, and some hemoglobin variants are similar in their electrophoretic mobilities, and therefore it will be difficult to differentiate them. Then they have some hemoglobin variants in which the amino acid substitution is internally, not externally, and therefore they may not separate from the rest of the hemoglobins, other hemoglobins. Those that have an amino acid substitution, that has no effect on the overall charge will not separate by electrophoresis. But there are other methods of separating these hemoglobin variants, and they include acid agarose gel, isoelectric, isoelectric focusing, and high performance liquid chromatography. These are more expensive and they are not regularly used in most labs in Trinidad. The high performance liquid chromatography, or HPLC, is a very fast, sensitive, largely automated method which requires minute quantities of sample. That is one of the advantages. It has become the method of choice in many labs, particularly for large-scale population testing, such as neonatal testing programs to determine if the neonate or the recently born offspring has any abnormality in their blood, hemoglobin specifically. And we'll have the lab techs again showing you what the procedure is like.
Okay, so this is lab 9, as Dr. Campbell was saying. This is our hemoglobin electrophoresis setup here. We have our Titan acetate, cellulose acetate plates that we use. We soak these for a minimum of 20 minutes using the super heme buffer. This is basically how we put it on the sample racks. We place it in the empty container and then place this on top. This have an aperture, a small hole there, that we fill this with the super heme buffer, and it gently runs down on the plates, eventually reaching up to the level of the plates and soaking them equally or evenly. We don't ever just immerse it into the buffer. So that's the first thing we need to do when we're setting up. Let our plates soak in advance using the cellulose acetate plates. Then we obtain our samples and our hemosylate reagent. This is used to lyse the blood cells, because basically we want the release of the hemoglobin inside of the red blood cells, because that is what we are going to analyze. Right? We have our chamber that we actually run our plates. We put our plates and we get our reaction, whether it's negative or positive, and our bands are pulled to the direction that they are charged by the electrophoresis power supply. Then we have our stains. Right, so that is the process. We set up our sample, we put it on the chamber, and we stain them, then we dry our plates. So now we have our whole blood samples. Again, we're using whole blood. Then we have our tubes. So these tubes are basically to do the lysing, as I spoke about before, to release the hemoglobin from the red blood cells using our hemosylate reagent. The dilution is basically a 1 part whole blood to 3 parts hemosylate. So we're going to use 75 microliters of the hemosylate reagent to 25 microliters of the blood sample. So we transfer the hemosylate reagent to our tubes, 75 microliters. And we use our 25 microliters of the blood sample. So 1 part to 3 parts hemosylate. A little mix. So now we have our diluted whole blood samples. So as you can see, it's immediately starting to lyse the sample. It's becoming like a bright cherry red in color. So already the hemoglobin is being released. Then we want to put it in the wells. This is where we place the blood sample and take them up to put them onto the plates. So this here is a hemoglobin electrophoresis control sample. We have the A band, F band, S band, and the C band. In all laboratory testing, we need controls. In this case, we always put our control in well 1, position 1. So therefore, when we are reading our plates, we can read it against our controls, and we'll have reference lines, reference marks where the hemoglobin, the different hemoglobins will migrate to on the cellulose acetate plate. So we're going to put 5 microliters of the sample into the wells. So we can pay close attention to the technique. We dispense the sample on one end of the well and just place our pipettes around like that. And do it again. So control in position 1. We're going to use a sample. Dispense. Okay, so we're now going to transfer our samples onto our cellulose acetate plates. Make sure you always have distilled water on hand. This is to prevent the plates from sticking to the surface that you're going to use. In this case, we're using a Petri dish. We're really supposed to be using an application cathode. So, we're going to put 1 drop of the distilled water. So now we're going to prime our Zip-Z sample applicator. Going to put it facing forward like that onto the wells. Going to hold firm and go down for 3 to 4 times. 1, 2, 3, 4. Then we're going to put it onto our filter paper. Okay. This step is very important to make sure that we have equal amounts of the sample onto the plates and to get rid of any excess. Then we take out our plates. We blood dry them. This process here now is to obviously get rid of any excess buffer on the plates, and also to be able to see which side is our acetate side and our glossy side. This is the side that we're going to label, not to mistake the acetate side from the glossy side. So we're going to label this plate, plate 1, using a China pencil. Then, glossy side, we're going to put a drop of distilled water. This is for the plate not to stick to the surface, and we're going to flip it over to get our cellulose side up. Now we're going to apply the sample. So this is what it should look like. Good. So class, we're going to prepare the electrophoresis chamber. So this is it here. So class, this is the preparation of the electrophoresis chamber. One of the things I want to emphasize is the actual removal of the cover. So the manufacturers place something here, and that's where you're going to put your thumb, and your index finger goes underneath. Able to remove. The buffer goes to the outer chambers, and 100 milliliters of the buffer. Goes into both sides. I'll show you how the actual wicks are placed on one side. So we just have to repeat it to the other side. So we place the wick into the buffer. Ensure that the bottom is in solution, and we fold. Okay, so we have our plates. When we place a sample on it, we need to put it face down with the samples facing more to the negative side of the chamber. It's very important. Face down. So acetate side face down, facing the negative side. Very important. Then it's just a technique I like to do. Put the slides on top as an anchor. Close. And then we set. The manual instruction says that we adjust the voltage to 350 for 25 minutes, and that's basically the machine settling to get to the correct voltage. And then we leave this for 25 minutes. Then we go on to staining. So we're going to remove the slides from the chamber. I'm going to stain. Try your voice, right? The staining procedure has to go into Ponce US for 6 minutes. Right, so class, it's about to time. So from this, after the stain, we're going to the acetic acid. 3 stations of that, 2 minutes each, and that part of the staining procedure is to de-stain. So after the 3rd acetic acid, place it in methanol
to dehydrate the slide. 2 minutes again. 2 methanol. So after the second methanol, we're going to put it in permaclear for 3 minutes.
Okay, so this is how the HBEP turned out. The white background that you're seeing is because of the clear rate, so that is a limitation of the lab. Your clear rate needs to be freshly made up, and therefore the entire plate or the acetate will be cleared up, and when it dried, the plate itself will be clear or transparent, and then you read it against a white background. But in this case, we can still see the bands. We can still see our control bands, which is in first position, and we can see where our sample bands migrated to. So remember, I told you before that your control bands are A, F, S, and C. Okay, so we have our control band on top, our reference band, AFSC, which is AFSC here, and we read our sample against the control sample. So patient 1, we have a A band only, which falls under the control A, which is interpreted as A plus A2. Patient 2, we have a C band only under the control C, which is interpreted as CEO. And patient 3, we have an S band only, falling directly under the control band S. SDG, it is interpreted as. So you can continue interpreting the other bands for the other patients.
One point is that to quantify, we cannot quantify the type of hemoglobin by just looking at the bands. We could say, well, one band looked a little heavier than the other, and therefore there be more of that hemoglobin. But there's an instrument called a densitometer, which we can attach to the plate, and you can read the quantity, or you could determine the quantity of each of the different hemoglobin bands. But we don't have that here at the moment.